Hello everyone, welcome to ITLS Academy. This is your mentor Dr. Shamaita Basu, and in this session we will be covering the second part of the topic molecular diagnostics. So in the last topic I think we finished with the structure of DNA, the different bases, how the complementary base pairing happens, what are the different kinds of bonds present between the bases, the different purine and pyrimidine bases, and the anti-parallel and complementary nature of the DNA helix. So in this session we will be starting with the concept of gene. Now what is gene? Genes are present on the chromosome, and DNA are many such chromosomes together. So DNA is basically the highly condensed form of chromosome, and chromosome is basically the highly condensed form of gene. And what is gene? It is a unit of inheritance, and it carries the information for a polypeptide chain and for the structural RNA molecule. So polypeptide chains mean basically, to put it simply, to put it in a simple explainable language, any gene will code for a particular protein. So that protein can be an enzyme in nature, it can be a hormone, it can be a chemical messenger, or it can be as simple as a RNA molecule from where the protein will be formed by translation. Okay, because that is the central dogma. From DNA will be formed, that is replication. From DNA RNA will be formed, that is transcription. And from RNA finally the protein product, the desired protein product will be formed, which is translation. Okay, so this is basically how a particular structure of a gene looks like. So this is basically the structural gene, this is the promoter which is always upstream of the structural gene, and this is the downstream region. So always remember, I think this is something I've already talked about when I was talking about the operon structures. So basically downstream means right hand side of the structural gene, and upstream means left hand side of the structural gene. Okay, so the promoter will always be towards the 5' end, and the downstream part will be towards the 3' end. Okay, now moving forward to nucleases. What are nucleases? They are enzymes which will be cleaving the different nucleic acids, which can be DNA, which can be RNA. So here we will be focusing on cleavage of the DNA strand, or just assume it as a structural gene. Okay, whether DNA or RNA. So we have a 5' exonuclease and we have a 3' exonuclease. What will they do? They will, nuclease means anything, any term which is ending with the term ase, it means it's an enzyme. Okay, so what kind of enzyme? Nuclease means it will be cleaving or it will be breaking down nucleic acid. So 5' exonuclease, exo means outside, so it will be breaking it towards the, or cleaving the strand towards the outside, not towards the inside. If inside, then it's an endonuclease. If outside, then it's an exonuclease. Okay, so here see, the cleavage is happening on the outer side of the 5' end. The product will be somewhat like this, and in this case the cleavage is happening towards the outer side of the 3' end, so the product will be somewhat like this. But in this case we are showing the endonuclease. So endonuclease will be cleaving towards the inward side. So see, here the cleavage is happening not towards the outside side but towards the inside side. Okay, now coming to the concept of restriction endonucleases or restriction enzymes. What are they? These are basically specific endonucleases which will be cleaving at particular sites which are known as the restriction sites. Okay, so they will recognize specific short sequences of DNA and cleave the DNA at or near the recognition sequence. So that is why I mentioned about the term recognition site or restriction site. Means specific sites will be there, and these enzymes will be cleaving only at those specific sites, not anywhere else. Like they will not go on and randomly go on cutting structures or nucleases at different sites. Okay, so the recognition sequences, how, like what will be the characteristics of this recognition sequences or recognition sites, or also restriction sites, same thing. So there will be usually 4 or 6 bases in length. So 4 or 6 bases means, let's say ATGC, uh, ATGC, or maybe ATGCTA, or whatever it is. Okay, so 6 such bases will be there, 4 or 6 such bases will be there, but there are some which are even much longer, like 5 or 8 or even 10 to 12 nucleotide bases in length. And these recognition sequences are always palindrome. Again, what is palindrome? I have already mentioned earlier. So palindrome means when read from left hand side to right hand side, or from right hand side to left hand side, it's the same. There's no difference. That means it's a palindromic sequence. Okay, or they are also known as mirror sequences. Okay. Okay, now what is a palindrome? They have defined a palindrome over here. So it's a sequence of DNA that is the same when one strand is read from left to right, or the other strand is read from right to left. Consists of adjacent inverted repeats. Okay. So continuing with restriction enzymes. So they have just given a nice example of a palindrome over here. So see, if we read this base sequence on this strand, on the upper strand, from the left hand side to the right hand side, GAATTC, and then this is the other strand, the complementary strand, and if we read this sequence from the right hand side to the left hand side, it will be exactly same as this sequence read from the left hand side to the right hand side. See, GAATTC, here also GAATTC. Okay, so that is what is meant by a palindrome. So restriction enzymes are basically isolated from bacteria. So different types of bacteria are there which are expressing different plasmids and hence thereby expressing different restriction sites and restriction enzymes. Okay. What are plasmids? Plasmids are extra chromosomal DNA, means DNA which is present outside the normal bacterial chromosome. So it's an extra DNA, you can say, and it plasmids, they have specific characteristic features, and they are most important is that they are self-replicating. They can replicate on their own. They do not need a separate cell machinery for their replication. That is the most important or unique feature of plasmids. Okay, and they also play a very important role in in transmission of antibiotic resistance from one bacterial strain to the other. Okay, so since these restriction enzymes are isolated from bacteria, they are named also by the name of the bacteria. So let's just see. Derive names from the bacteria. Genus is the first letter, which is capital, like the first letter of the genus is capitalized, and then the second and third letters will be coming from the species name, and they will be written in small cases. Okay, and there will be some additional letters also which will be coming from the strain. Roman numerical designates different enzymes from the same bacterial strain in numerical order of discovery. So, say for example, we have discovered say 10 restriction endonucleases from the same bacterial strain, say for example E. coli. So, how will we name them? We will be using Roman 1, 2, 3, 4 for naming them. Okay. So, just an example, see Eco R1. So, Eco R1 stands from Eco, E, first letter of the scientific name, genus name, and eco, first two letters of the species name. So that's how it's made, eco. And R1, R stands for restriction and 1 stands from Roman R. Okay, so see, they have explained also. E stands for Escherichia, co stands for coli, R stands from R stands for not restriction enzyme, but R stands for R strain. Which type of strain it is isolated from? It is isolated from R strain. Okay, and then we have 1, which is the Roman 1, means it's the first enzyme to be discovered from the R strain of Escherichia coli. Maybe there after then eco R1, R2, R3, many other significant enzymes have been discovered from the same R strain of E. coli. Now coming to something called hybridization. What is it? Nucleic acid hybridization is the formation of a duplex between two complementary sequences. So say for example, there are two complementary sequences and we need to form a bonding or an association between the two complementary sequences. So that is what is known as nucleic acid hybridization. Hybridization, the word comes from the word hybrid. Okay, means the end product of hybridization will be a hybrid. So there can be intermolecular hybridization, means it is occurring between two polynucleotide chains which are having complementary bases. Means two polynucleotide chains means it can be between two DNA molecules, it can be between two RNA molecules, or it can also be between a DNA and a RNA molecule. Okay, so that is intermolecular hybridization, because you are hybridizing between two polynucleotide chains. And what is annealing? Annealing is another term which is used to describe the hybridization of two complementary molecules. Annealing means basically jodna. Okay. So basically it's a another, you can say, synonym for hybridization. Okay. Now coming to the concept of denaturation and renaturation. What do you mean by denaturation? When a particular structure of a particular compound, here we are talking about DNA, RNA, but basically denaturation term goes well with protein. Okay. So denaturation means the normal configuration or the normal structure of that compound will be disrupted. It will be damaged. That means it's denatured. And what is renaturation? When from that denatured state the normal structure will be revived, the normal configuration will be derived, will be revived. That is what is renaturation. So renaturation will occur only when denaturation has already occurred. Then only the question of renaturation will arise. Okay, so renaturation means revival of a denatured compound or denatured configuration to back to the normal configuration. That is what is renaturation is. So say for example, here I have shown a double-stranded DNA. So see, three such double-stranded DNA molecules are being shown. One strand is shown in yellow color and the other strand is shown in light bluish green color. So this DNA molecule, say for example, is denatured. How it will be denatured? When it will be exposed to high temperature, usually more than 80 to 90 degrees Celsius. Okay, so that is the temperature that is required to denature the double helical structure of the DNA. Okay, so as a result of denaturation, we can see what is happening. The strands are getting separated from each other. Why? Because the hydrogen bonds between these strands of DNA, they are getting broken down due to the heat, because they are heat labile. They can no longer resist the heat or tolerate the heat, and hence the hydrogen bonds are getting dissociated and the strands are getting separated from each other. The bonds between the strands are getting separated. Okay. So after that, what will happen? So as a end product of denaturation, what we are getting? We are getting the single stranded DNA. After that, there will be base pairing. So see, the renaturation process has started automatically on its own. So after some time, as the temperature automatically decreases, say for example to 50 to 60 degrees Celsius, the DNA strands will automatically start annealing with each other. They will automatically start hybridizing or forming complementary base pairs with each other. So the renaturation process will already start from this step, that is initial base pairing. So after that, the proper renaturation will occur and we will get the renatured DNA, which is exactly same as the double-stranded DNA that was there before we did denaturation. Okay, so renaturation is just a revival of the original structure, which can be possible only after the DNA has been denatured. Okay. Now talking about probes. What are probes? Probe is nothing but a nucleic acid, or you can say a sequence or a part of a nucleic acid which will be labeled with a marker which allows identification and quantitation, and it will hybridize to another nucleic acid on the basis of base complementarity. Very important. So say for example, we want to localize or we want to identify a particular sequence, say for example ATG. Okay, so this particular sequence is there on a particular DNA strand and we want to identify this sequence for whatever reason it may be. How will we do it? We will synthesize a probe which will be exactly complementary to this particular sequence of the strand. So ATG is the sequence that we need to identify. So complementary to it means it will be TAC, isn't it? So the probe which we will get or the probe which we will synthesize will be having the sequence as TAC, just complementary to ATC. Okay, so now what will this probe do? Since it's complementary, it will easily go and bind with that particular sequence. It will easily go and hybridize with that particular sequence. And then how will we be able to identify it? Because we will be labeling this probe with a marker. Now this marker can either be a fluorescent marker. If it's a fluorescent marker, so it will be producing fluorescence, there will be a color change. So in that case, it will be known as a fluorescent probe. Or it can also be an enzyme in nature. So if it's an enzyme in nature, then it will be a one more step harder it will be the process to achieve. Why? Because we will have to add a substrate. on which the enzyme will react, and then the product will be formed, and that product will lead to a color change which can be detected either visually or quantitatively. Okay, how will we detect color change quantitatively? Using a colorimeter or a spectrophotometer, which will be absorbing the change in the light wavelength, and hence they will be giving a measurement, a value. Anything which we are measuring quantitatively means we need a value. We need a numerical value. That means it's a quantitative measurement. And qualitative means we only have a picture or we only have a visual observation. That means it's qualitative. We are not dealing with any values or any numericals. Okay, that is the basic difference. So whenever we are performing any experiment, it is always suggested, or it is always preferable that we go for a quantitative estimation rather than a qualitative one. Okay, now there are different types of labels. Oh, okay, a very important thing that I forgot to mention, besides the fluorescent and the enzyme labeled probes, we also have the most important one, that is the radioactive probes. Okay, so there we use different radioactive metals, like we have potassium, 32 potassium, then we have 35 sulfur, we have 14 carbon, and we have deuterium, that is tritium, that is 3 hydrogen. So these are some of the radioactive metals or radioactive components that we can label the probes with to get the satisfying signals as a means of measurement. Okay, and we can also go for fluorescent probes, which I already mentioned. Fluorescent probes are usually used in case of fish is again something I have explained in the initial sessions of this course of medical biotechnology, that is fluorescent in situ hybridization. Okay, or fish, that is a very important aspect or very important basis of different diagnostic techniques of like dealing with different sections of medical biotechnology. If you have forgotten, you can always go and watch the video. Okay, I think it was in the starting 1 or 2 sessions I had discussed about fish. Okay, and we can also use biotinylated labels, where we can use different antibiotic like chemicals or chemical components rather, like avidin and streptavidin. Okay. Okay, now coming to polymerase chain reaction, again something I think I have talked about in the initial 1 or 2 sessions of this topic of medical biotechnology. So polymerase chain reaction, basically it stands for, chain reaction means something which will be going on and going on and going on, isn't it? It's occurring in the form of a chain. So basically usually we go for 30 cycles of amplifying a particular DNA sequence. So basically polymerase chain reaction is mainly identical or mainly important or mainly significant in case of forensic science, I would like to say, because what happens from the what is forensic science? Forensic science is dealing with the different crime scenes or different criminal scenarios. So what happens whenever a person is committing a crime, some or the other kind of proof he or she has to leave behind. Okay, so in from most of the crime scenes, different DNA samples are collected, either in the form of fingerprint or in the form of nails or hairs or teeth. Okay, so in many cases, what happens, the amount of evidence that is collected is very, very less, very, very small. Okay, to further carry on with that evidence, or to further come up with something evident or something fruitful with that particular specimen. So what we need to do is, there's an urgent need that we can amplify that small portion of DNA to get a larger amount of DNA and thereby deal with that DNA for further investigations. Okay, so there comes the necessity of PCR. Okay, not only for forensic sciences but also for different diagnostic techniques, PCR is very, very important. So for example, in case of covid pandemic, we all of us, we have like experienced the COVID pandemic. All of us have a fair amount of idea about what it was. So in case of COVID, we saw that PCR, RT-PCR was necessary for confirmatory diagnosis of COVID. Okay, different rapid tests were there, but in many cases it was seen that the rapid tests are coming positive, but actually after RT-PCR we are seeing that the patient is actually negative, or the other way around. The rapid tests are coming negative, but after doing RT-PCR we are seeing that the patient is actually positive. Okay, so that is where comes the importance of PCR. because ultimately here we are dealing with DNA or RNA sequences. Okay, so it has to be confirmatory in nature. So basically it is the in vitro enzymatic, in vitro means which is outside the body under lab conditions. Enzymatic synthesis and amplification of specific DNA sequence. This term specific is very important over here, because we are dealing with particular DNA sequences which are of our concern, which we need to study with, which we need to amplify for further studies. Okay, so in vitro, why? Because normally inside our body that specific sequence would have multiplied, would have replicated, isn't it? But since we are collecting the sample from outside the body, from outside the microorganism, hence we need to do it under lab conditions. Okay, so PCR can amplify one molecule of DNA into billions of copies in few hours. As I mentioned, 30 cycles. So in 30 cycles, basically it increases exponentially. Okay, so from 2 you will get 4, from 4 you will get 8, from 8 you will get 16, from 4 you will get 16, from 16 you will get 32, like that. So exponential multiplication is there, 2 to the power n, where n is the number of the copies. Okay. So coming to applications of PCR. Okay, so the applications will include detection of chromosomal translocations, uh, like particularly amplification across a translocation sequence. It will also include chromosome painting. It will include detection of residual disease. Residual disease means a disease which is not actively present in the patient, but still the patient is suffering from the disease. That means it's a residual disease. So such diseases are usually very tough to be detected since it's in the residual form. Okay, so there also PCR plays a very important role. And also infectious diseases, example I have already talked about, COVID. And forensic science, again example I have already talked about. HLA typing, human leukocyte antigen typing, that is also a very important application of PCR. Okay, and detection of loss of suppressor genes or suppressor genes, for example loss of heterozygosity. So what are suppressor genes? Suppressor genes are those genes which are responsible for suppressing a particular characteristic. Okay, so if that suppressor gene is basically, suppressor genes are very important for preventing cancer, preventing different types of cancer. Okay, so in order to detect that loss, whether the suppressor gene has been lost or not, for final diagnosis of cancer in the patient, again PCR application is very important. Okay. Okay, now coming to genome literacy. What is genome? The entire DNA of an organism. That is the basic difference between gene and genome. Because gene means single. Many such genes can be present in a single chromosome. And many such chromosomes will go and form the DNA, isn't it? So genome means the entire DNA of an organism, and we all know that humans are diploid organisms, means they're always having chromosome pairs. Total 23 pairs of chromosomes are there in the human body, means 46 chromosomes in total. About among them 22 pairs are autosomes and one pair are the sex chromosomes, which is XX in case of female and XY in case of male, normal male and female. Okay, there are various other conditions also which I have already talked about, like there is trisomy. Trisomy 13 is basically autosomal disorder, but sex chromosome disorder will include disorders like Turner syndrome, Klinefelter syndrome. Okay, so those are the exceptions, but normally dekha jaye, so it's either XX in female or XY in male. Now, how many base pairs are there per diploid genome? 6 into 10 to the power 9. And haploid genome means one set of chromosomes. So haploid number is denoted by n and the diploid number is denoted by 2n. These are the things that all of us know very well. And what is chromosome? Chromosome is the structure found within a cell nucleus consisting of a continuous length of double-stranded DNA. Okay, so continuous length means double-stranded DNA will be continuing and that will be forming the chromosome, which will be present inside the nucleus. Okay, and humans will be having, as I mentioned, 22 pairs of autosomal chromosomes. Autosomal chromosomes means they will be present in all the cells of our body. Okay, irrespective of whether they are the gonads or the normal body cells. But sex chromosomes will be only one pair, two in number, XX or XY, which will be present only in the gonads. Okay, gonads means ovum or egg or ovary in case of female, and sperm in case of sperm or semen in case of male. That is the gonads. Okay, now talking about the Human Genome Project. Something I will be also talking about in the when I will be talking about human gene therapy. Okay, so the Human Genome Project, I think it started in the year 2000. Uh, it it ended with the conclusion that the human body contains about 40,000 genes, like with all the different variations that are there. Okay, and basically the Human Genome Project was a huge success. It spoke about a language of molecular fingerprints, because once we get to know about all the different genes that are present in the humans, it will be very easy for us to detect and to to pinpoint different crimes, different criminals, different types of diseases, different types of variations, genetic predispositions. These things can be easily identified once we know a fair picture about the different genes, isn't it? So basically it's a language of molecular footprints, it's fingerprints. Basically it's a language of molecular biology science. Okay, and gene expression is another language of complexity. Gene expression is something different, and Human Genome Project is something different. So the Human Genome Project was famous because it gave us a broad perspective about the different kinds of genes that can be present in the human body. Okay, so it started in the year 1990 and it ended in the year 2003. Okay, so last year we celebrated 20 years of the Human Genome Project. Okay, okay, now coming to genome mapping terms. So while talking about mapping terms, we need to know what is locus. Locus is the position of a gene on a chromosome. Okay, so where on a particular chromosome a gene is present will be the locus of that particular gene. Next is allele. What is allele? Allele is nothing but a copy of a gene. Alternate form of DNA at a specific locus on the chromosome. Each individual will be inheriting two copies of DNA. So there will be, for a particular gene, there will be two alleles. One will be derived from the mother and the other will be derived from the father. Okay, so two alleles will be there for a particular gene or a particular uh, on a particular chromosome. Okay, and some of the alleles will be homozygous, some of the alleles will be heterozygous. Now these are the things we already know. Homozygous means when the two copies will be exactly same, identical, and heterozygous means when the two copies will be different. So this will depend on probability, isn't it? Whether the alleles we are inheriting, whether they will be homozygous or they will be heterozygous, it will be depending on probability, and the probability will be determined by the genetic makeup of our parents. Okay, okay, now coming to genetic variation. What is it? Most genes have small sequence differences between individuals, which occurs every 1350 base pair on an average. So, with at a difference of 1350 base pair, the genes will be having sequence differences, which will be very minor or small. Some of these polymorphisms may affect how well the protein works, how well the protein interacts with another protein or substrate, and the different gene forms containing polymorphisms will be containing alleles, will be known as alleles. Exactly what I mentioned. So allele is basically same form of a particular, two forms of a particular gene. Mutation detection, how it can be done? It can, what is mutation? I've already mentioned in the previous sessions also and in this session also. So it can be done by sequencing DNA. It can be done by hybridizing methods, which will be including blotting and application of chips, magnetic chips on the DNA. And it will be also including restriction enzyme polymorphism, which is also known as RFLP, restriction fragment length polymorphism. Okay, where we will be using the different specific sequences where the restriction enzymes will be going and cleaving at those particular restriction sites. And we can also go for SNPs, again something I've already talked about in one of the earlier sessions of this course of medical biotechnology, that is single nucleotide polymorphism. So it can also be utilized as a method for detecting mutation. So I think that's it. With this we complete with the topic of molecular diagnostics. Next topic I think is protein therapeutics, which we will be starting shortly. Till then, thank you, goodbye. Please like, share and subscribe ITLS Academy.